Hey guys, it's Medicosis Perfectionalis where medicine makes perfect sense. Let's continue our biochemistry playlist. In previous videos, we talked about enzymes, we talked about competitive and non-competitive inhibition, we talked about cooperativity and the Hill's coefficient, and even biosignaling. Today, we will get to the basis of everything, no pun intended. It's time to talk about your deoxyribonucleic acid, the genetic code of your body. Please try to watch the videos in my biochemistry playlist in order. What's the fuss about? Let me give you an analogy. The genetic code is like a computer code. You can see the hardware. That's easy. However, you cannot see the software. Similarly, DNA is not just sugar, nitrogenous base, and phosphate. It also carries information. Just like when you send an email from person A to person B, telling person B, hey, go meet Mr. X in New York. He is able to understand you. The zeros and one of the computer code and the letters of the alphabet are not just symbols. They carry information that can be understood by the recipient. The same is true for your DNA. The DNA inside the beta cell in your pancreas will tell the cell, hey beta cell, go make some insulin. This is a message that carries meaning that can be translated into actual insulin release, which helps lower your blood sugar by pushing glucose into the target cell. Why does the target cell need glucose? To make energy. Oh, that makes sense. The building unit of your body is the cell. A group of cells will make a tissue. Bunch of tissues, organs. Bunch of organs, systems. And the system will perform the body function. Inside that lovely cell, we find the nucleus, which contains your DNA, which is your genetic code. Is the nucleus the only place in the body that has DNA? No, it's the main one, but it's not the only one. Your mitochondria do have DNA. The DNA in your nucleus was inherited half from mommy, half from daddy. But your mitochondrial DNA only came from your mommy because your daddy's sperm went deep inside into the ovum but left his middle piece and his mitochondria outside. So the mitochondrial DNA that you have came from your mommy, from the ovum, not from the sperm. And if you are a plant watching me, well, you do have DNA in the chloroplast. What does the word plast mean? Synthesis, formation, like plastic surgery. That's why we call plastic, because it's synthetic. Where does DNA live? It lives in the nucleus. Okay. How come DNA does not leave the nucleus and go outside to tell the pancreas to make insulin? That would be a disaster. If DNA left the nucleus and went outside, it will be degraded so quickly it will make your head spin. So DNA will stay inside. And then send a messenger, like messenger RNA. And this messenger is capable of leaving the nucleus through the nuclear pores and going to the cytoplasm. And then we can translate the message that we got from the code. And when you translate the message, you get meaningful proteins. Here's your lovely DNA. It's anti-parallel. Never ever forget that phosphate is at the 5 prime. Phosphate 5. Who is at the 3 prime end? Just OH. The building unit of your body is the cell. Each cell has a nucleus. The nucleus contains 46 chromosomes, assuming that this is a somatic cell. The chromosome contains your DNA, compacted and wrapped around itself, or around special proteins known as histones. DNA stands for deoxyribo, that's my sugar, nucleic, nitrogenous bases because they have nitrogen, and because it's in the nucleus, acid. What's the function of that DNA? Well, the DNA codes for protein. It sends meaningless messages that can be translated into meaningful proteins. DNA is made of sugar, nitrogenous bases, and phosphate. What kind of sugar? Deoxyribose sugar. What kind of nitrogenous bases? Well, it could be adenine, guanine, thymine, or cytosine. Adenine and guanine are known as purines. The mnemonic is purines are pure as gold. As for thymine and cytosine and something else present in the RNA called uracil, those three are pyrimidines. The mnemonic is cut the pyramid. cut, C, cytosine, U, uracil, T, thymine. The pyramid, pyrimidines. And just like the base of the pyramid, it could be a triangular base for that pyramid, or it could be a square base for the pyramid. Either way, it's just one ring. So if you have one ring, you are a pyrimidine. If you have two rings, you are purine. My DNA is deoxyribose sugar, nitrogenous bases, and phosphate. You may forget your name, but never forget this. Very important. Your DNA is super duper long. A piece of it is the gene. How many genes do I have? More than 10,000? DNA is sugar, nitrogenous base, phosphate, sugar, base, phosphate, sugar, base, phosphate. What kind of sugar? Deoxyribose sugar, okay? Ribose, as you know, is a pentose sugar, which means it looks like a pentagon with 5 sides and 5 corners. Next, the nitrogenous bases. What kind of bases? Well, adenine, guanine, thymine, cytosine. Adenine and guanine are purines, double ring, but thymine and cytosine are pyrimidines, and the pyramid has a single ring. Pure as gold, and cut the pyramid. Cytosine, uracil, and thymine. Okay, I understand that they are bases, but why nitrogenous? Because each one of them has nitrogen. Duh. And then we have the phosphate. Sugar plus nitrogenous base equals nucleoside. Nucleoside plus phosphate equals nucleotide. There are two mnemonics here. You can say nucleotide has phosphate, or you can say that nucleotide has triple components, a triad, a trilogy. Sugar, base, phosphate. DNA versus RNA. The DNA has deoxyribose sugar. RNA has ribose sugar. Make no mistake about it, both of them are pentose sugars, like a pentagon. But hey, Medicosis, what's the difference between ribose and deoxyribose? The name has the answer. D means without, like stabilize versus destabilize, without stability. So this one has no oxygen, without oxygen. Deoxy. In which part? Carbon number 2 prime. Look at that. This is an O. Oh, this one has an oxygen at number 2. But look at that. There is no oxygen. Where is the oxygen? The moment you ask where is the oxygen, we're talking about deoxyribose sugar. My wacky mnemonic is look for O2, oxygen, at carbon number 2. And I understand that these are not 2 oxygen, it's just 1 oxygen. I get it. The mnemonic is imperfect. Come on. When you add a sugar to a nitrogenous base, what do you get? Nucleoside. What kind of sugar are we talking? Ribose. Oh yeah, I can find the oxygen. How about the base? We'll say adenine, for example, and then add ribose to adenine. Before you know it, it's adenosine. Sine will rhyme with side. So when you see adenosine, you know it's a nucleoside. Look at that, it has 2 rings. Why? Because adenine is pure as gold. Purines have 2 rings. Why is it nitrogenous? Look at all of those doozy nitrogens. And then let's join number 9 from the nitrogenous base with number 1 from the ribose, and before you know it, we have a covalent bond between them, and this is now called adenosine. Notice something. The numbering system here does not have a prime. To differentiate, we added a prime here. So this is 1 prime, but this is just 1. This is 2 prime. This is just 2. So how do I remember this? I just say prime sugar. The sugar has the prime. So ribose plus adenine equals adenosine. That's the nucleoside. If you add ribose to guanine, however, you get guanosine. If you add deoxyribose sugar to thymine base, what do you get? Deoxythymidine. How about adding deoxyribose to cytosine? Deoxycytidine. How about adding ribose to uracil? You get uridine. How about adding deoxyribose to uracil? You get deoxyuridine. Sugar plus base equals nucleoside, but sugar plus base plus phosphate equals nucleotide. If you added 1 phosphate group, we'll call it adenosine monophosphate. If you added 2 phosphates, we'll call it adenosine diphosphate. If you added 3 phosphate, which is my favorite, adenosine triphosphate. Why is that your favorite? It's a source of energy for your cell. Why? Because if you break those 2 bonds, what do you get? very high energy that you can take advantage of for your own sake, to perform your cellular functions, or to activate your sympathetic nervous system while running from a tiger. Well, you will run a lot, requiring too many ATP molecules.
But hey, Medicosis, I don't get it. Why do they have tons of energy there?
Because it's the repulsion force between the negative phosphate, etc., that creates too much repulsion. Too much repulsion, potential for energy. And that's why ATP has way more energy than ADP, which has more energy than AMP. Another point to remember, again for the gazillionth time, the phosphate is at the 5' end. Phosphate 5. The phosphate is at the 5. Phosphate has a P', 5', which means it's attached to the sugar. Hashtag prime sugar. Phosphate is not attached to the base. It is attached to the prime sugar. Let's add some French to the mnemonic. This phosphate has a negative charge. And the phosphate is full of energy. That's the story behind ATP. If you remember my physiology video titled Muscle Metabolism and Sources of Energy, we talked about how your muscle gets energy during rest. It was from the sodium-potassium ATPase pump. What do you think ATPase does? It breaks down ATP. When you break down ATP, boom, you release energy. A reaction that releases energy is coined exothermic.
It releases heat.
Here is the lovely ATP molecule again. Here is my sugar. Here is my base. Here is phosphate, ribose sugar, adenine base, and 3 phosphates. Look at this lovely bond. It's between two negatives. High energy, high repulsion. This one also has high energy. Why? Negative repulsion. High repulsion equals high energy. But look at this doofus. Ordinary energy. Because there are no two negatives here. Get it? That's why ATP is amazing. But AMP, no one cares. Pause and review. When you break down ATP, you release energy and phosphate. Remember, adenine was here, and there is a bond, and then phosphate, and then another bond, and then phosphate, and then another bond, and the last phosphate. When you break that bond, you release the energy, exothermic, and the phosphate will go to hell. You're also releasing phosphate. Who's left? ADP, adenosine diphosphate. Let's break another bond. Boom, energy is released and phosphate goes to hell. And you're left with adenosine monophosphate. The rule in chemistry is that most of the time, when you break a bond, it's an endothermic reaction. But making a bond is an exothermic reaction. You want an exception? ATP is an exception. We're breaking a bond, but releasing energy. We're breaking a bond, but exothermic reaction.
How do we break down the ATP into ADP and release the energy?
ATPase, an enzyme that breaks down ATP.
How do I break down ADP and release energy?
Also ATPase.
How did my muscle get ATPase?
It was here, in the head of the myosin, which is a contractile protein. Please refer to my biology playlist.
Okay, Medicosis, now I get it that ATPase is gonna break down ATP and release energy. How much energy are we talking?
12,000 calories. 25% will be consumed as the actual muscle work, and the rest is heat.
Isn't this inefficient?
Well, if you ask an engineer, yes. But if you ask Lionel Messi, this heat is very important. That's why Messi keeps warming up before the match. He knows what he's doing. This heat is providing an optimum temperature for the muscle enzyme activity. All of what I've said is how the muscle gets energy during rest. But now let's exercise. When you exercise, you will contract your muscles, squeezing the blood vessels between your muscles, decreasing the radius of the blood vessels, decreasing blood flow to the muscle. And if you do this a lot with strenuous exercise, there is decreased oxygen delivery to the muscle. The muscle will run out of aerobic and will turn to anaerobic metabolism. Let's review. This is glycolysis, does not need oxygen. It can happen in an anaerobic condition. But then the oxidative phosphorylation requires oxygen, of course. So during strenuous exercise, in the first, let's say 10 seconds, your muscle is using the ATP that was already stored there, and the muscle is remaking ATP from ADP thanks to creatine kinase. Why is that important? Because that ATP can be used for the next step. You'll need ATP for anaerobic metabolism. And then after a minute, if you're still exercising, well, the muscle has to go to the big guys, the TCA and the electron transport chain, to provide you with 38 molecules of ATP for each molecule of glucose utilized. Back to DNA, I need a deoxyribose sugar, I need a nitrogenous base, and I need phosphate at the 5' end. Phosphate is attached to the sugar. Sugar and phosphate are on the outside, but the nitrogenous bases are on the inside. My lovely DNA is double helix, anti-parallel. 5' to 3' here, but here the 5' is there and the 3' is up here. Moreover, your DNA has polarity. Here is at the North Pole and the South Pole, so to speak. Remember that the phosphate ends NO negative. The negative will attract the positive from the hydrogen, and therefore you have OH. So you have OH on this side and OH on this side. That's polarity. Next, complementary base pairing. A binds with T and C binds with G via hydrogen bonds. Very important. Those two were covalent bonds. Between the sugar and phosphate, covalent. Between the sugar and the base, covalent. But between the two bases, hydrogen bonds.
How many?
2 hydrogen bonds between A and T and 3 hydrogen bonds between C and G. That's why Cg is more stable, because it takes more energy to break 3 hydrogen bonds than the energy it takes to break just 2. Which means if you have 3 hydrogen bonds, you are more stable. That's why the greater the amount of Cg that you have, the greater the stability. My mnemonic is the greater the GC content, the greater the stability. GC, stability. In subsequent videos, I'll tell you that the primer has high GC content. You know why? For extra stability. The centromere between the 2 sister chromatids has high CG. You know why? For stability. The telomeres at the end of the DNA, what do they have? High GC content. Why? For stability, so that they do not unravel. Another important point. We read the DNA from the 5' to 3'. We write it from the 5' to 3'. How it gets replicated is from the 5' to 3', and transcribed from the 5' to 3'. And don't forget that phosphate is at the 5', and the 5' comes first. Now let's play the game of names. Here are the nitrogenous bases. Adenine is a base. When you add sugar to it, it becomes adenosine or deoxyadenosine. Adenosine is in the RNA, deoxyadenosine is in the DNA, deoxyribonucleic acid. So far we have nucleosides. Let's make them nucleotide. Adenosine plus 1 phosphate equals adenosine monophosphate, which is a nucleotide. 2 phosphates, adenosine diphosphate. 3 phosphates, adenosine triphosphate. All of this is RNA.
How about in the DNA?
Deoxyadenosine monophosphate, deoxyadenosine diphosphate, and deoxyadenosine triphosphate. Guanine, same thing. Nucleosides are guanosine in the RNA, deoxyguanosine in the DNA. You want the nucleotides? Guanosine monophosphate, guanosine diphosphate, guanosine triphosphate. In the DNA, deoxyguanosine monophosphate, deoxyguanosine diphosphate, deoxyguanosine triphosphate. Same exact thing with cytosine, but thymine is unique because thymine, the T, only exists in DNA and not in RNA, so there is no need to repeat it twice. It's always deoxythymidine in the DNA only, not in the RNA. That's the nucleoside. You want the nucleotide? Deoxythymidine monophosphate, deoxythymidine diphosphate, deoxythymidine triphosphate. And we can do the same story with the uracil, which is another pyrimidine. Please pause and review. You see this? That's your DNA double helix. It's very long, but your nucleus is very tiny. It's a microscopic structure. How come the nucleus, which is microscopic, holds my DNA, which is, let's say, 1 meter long? Well, we have to wrap it gazillion times around proteins known as histones. When the DNA wraps around histone, they are collectively known as nucleosome. So a nucleosome equals DNA plus histone. Keep wrapping, and now you have a string of beads. Wrapping. It gets darker and darker and more condensed, and now we have your chromosomes. You have 46 chromosomes in each somatic cell. Half of that in the ovum or the sperm. When the DNA is lovely like this and long and stretched out and not wrapped, it is called euchromatin, relaxed form. Eu means true. Why? Because I can truly work with this. If you want me to make RNA and therefore proteins, I gotta work with the euchromatin. But if you are condensed like this, you are different. You look different. Hetero means different. Chromatin. The chromatin looks different. It's not relaxed, it is condensed. Euchromatin appears lighter because it's less heavy, but heterochromatin is very heavy. Hetero, heavy, and appears darker in color under electron microscopy. Please pause and review the central dogma. When DNA replicates itself, it's called DNA replication. Make it into RNA, transcription of the message. If you go the opposite, reverse transcription. This is very important for HIV virus. RNA to proteins is translation. Translate the message from meaningless codons into meaningful proteins. Translation is the same thing as protein synthesis, protein formation, just like how your pancreas made insulin, which is a protein. The knowledge of DNA and chromosome has gazillion applications, such as in the field of genetics. Please watch my video titled
DNA is the genetic material.
You'll find it in my biology playlist, where you will learn about the locus, the gene, the allele, and the difference between genotype and phenotype. Also about 3 different experiments that proved to us that DNA is the actual genetic material in your body. All of these were discussed in my biology playlist. If you like this video, you will enjoy my kidney physiology course to learn about the proximal tubule, the distal tubule, the loop of Henle, the glomerular filtration rate, the renal blood flow, renal plasma flow, etc. Download it today at medicosisperfectionalis.com. If you want diseases, download my surgery high yields course. Trauma surgery, orthopedic surgery, vascular surgery, cardiothoracic surgery, and even ophthalmology. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my courses. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.